Hello everyone, welcome to the 7th video of the 100-day biopharmaceutics challenge. So today we are going to start with our next topic, that is going to be the basics of drug dissolution process. Okay, so let's see exactly what drug dissolution means. So from the name drug dissolution, it clearly mentions that how much a substance is going to dissolve, a solid substance will dissolve in a given solvent. So in case of our drugs, what actually that happens? So you have any solid dosage form, right? It is going to disintegrate. Upon disintegration, it is going to give us the solid drug particles. Now these solid drug particles will undergo dissolution till they reach the absorption. After then, they are going to cross the cell membrane, and then yes, we have the drug at the target portion, right? So basically the role of dissolution is very much important for us here, right? So for some drugs, what is actually happens? So for some drugs, the dissolution can be a problem, just like for some drugs the crossing the cell membrane is a problem. For drugs which are lipophilic in nature, for them crossing the cell membrane is not an issue because of the phenomena of like dissolves like. But for the hydrophilic drugs, the permeability can be a very much issue, right? So for crossing the cell membrane, it is going to be a rate limiting step for the hydrophilic drugs, right? Right? It is easy for the lipophilic drugs. It is going to be a rate limiting step for the hydrophilic drugs. Similarly, the dissolution is not going to be a problem for the hydrophilic drugs. It will be an issue, or we can say the rate limiting step will be for the lipophilic drugs. So anybody can ask you that dissolution is a rate limiting process in which of the following case of drug. So in case of the lipophilic drugs mainly, you have dissolution as a rate limiting step. In case of the hydrophilic drugs, you have crossing the cell membrane, the permeability may be a challenge. So what exactly is the dissolution, if I may ask? The drug dissolution refers to a process by which a drug is going to dissolve in a suitable solvent. Now, when it is going to dissolve in the biological fluids, for example, if you want to say, generally what happens, I'll give you one example by which I think most of your doubts regarding it will be clear. So suppose you have a drug, suppose you have a tablet here and you have a bulk phase here. Suppose you have a tablet here and you have a bulk phase here, right? So what is happening? It is going to release the drug. It is going to release the drug around. It's this section, right? We say that around the tablet, the concentration of drug is going to be high as compared to the bulk phase. So in this portion, in this we say that a film or a layer is formed in which the drug concentration is going to be higher in as compared to the bulk phase. So that in turns actually develops a concentration gradient due to which it is going to move more towards the bulk phase. Now this movement towards the bulk phase, this dissolution is actually governed by an equation which we call as Noyes-Whitney equation. Very important, students, Noyes-Whitney equation. So Noyes-Whitney equation basically gives the idea of the rate of the drug dissolution that is dependent on the surface area, partition coefficient, concentration of drug in bulk as well as the stagnant layer into your volume of the dissolution medium and the thickness. So based on this equation only your questions are directly asked. This is actually one of the theory of the drug dissolution process. Now we can clearly see with the previous diagram and this on what factors might the drug dissolution actually depends. So we have certain factors for you all that may affect the drug dissolution process. Number one is going to be solubility. Most definitely it is going to be a constraint for the lipophilic drugs. So for the drugs with the like dissolve like concept, the cell permeability is not going to be an issue, but the dissolution is going to be the rate limiting process. So yes, will be a challenge for these purely soluble drugs to actually dissolve, particle size, smaller particles have actually larger surface area, so you have a better dissolution there. Okay, next you have polymorphism. So it largely depends on the crystalline forms or the amorphous forms also. Salt formation, salt forms of the drugs are generally dissolved faster than the free forms. Okay, not only the properties of drug, the nature, the type, the size, the polymorphism, the crystallinity, the amorphism, whatsoever. Apart from this, we have a lot of formulation factors also, because ultimately your drug will be encapsulated in a particular carrier or will be made to fit in a particular formulation so that it reaches in a maximum possible concentration as possible. So number one, we have excipients. So based on what excipients are you using? Are you using disintegrants? Are you using surfactants, lubricants? All the excipients are going to have their own properties, own nature based on the type of formulation you are actually making. Are you making any controlled release formulation or what? Based on that, the dissolution will be impacted based on the nature of excipient also. For the dosage form, for example, solutions have this is the best order which you have I think previously read also. The solution will have the more ability for the dissolution, followed by the suspension, then capsule, then tablets in case of the dissolution rates. Coating. Now we all know we have film coating, we have enteric coating, sugar coating. So based on the coatings as well, you can actually help it to delay the drug dissolution. Why the delay in the drug dissolution is even required? I have just told you, in case of certain types of formulations, we need controlled release, delayed release, sustained release. So based on that, we actually decide the type of the coating. So that is going to alter the drug dissolution. So you have seen what are the basics of it and what can be the factors that can affect the drug dissolution process. Now apart from this, we have a lot of factors that may be result from the delaying the drug dissolution based on the patient profile also. For example, if you talk about the gastric emptying time, pH of the gastrointestinal fluids, and if the presence of food, that can delay the dissolution of certain drugs. Okay, so I think that's it for the basics of drug dissolution. In the next video, we will be covering the theories of drug dissolution, which will be a +1 to this topic. Okay. One more thing I think I would like to tell here, that from the dissolution testing only, we have certain apparatus that we have classified for you all so that you can learn them, because on these also the questions are asked. So based on the USP, IP and BP, for the USP we have basket, paddle, reciprocating cylinder, flow through cell, paddle over disc, cylinder over disc, and reciprocating holder. Now the images are slightly superimposed because these are the apparatus. Actually we have the basket here, then the paddle here, then the reciprocating cylinder, then the flow through cell, then the paddle over disc and cylinder over disc and reciprocating holder. Based on this order only you have to remember, you cannot jumble up these this order. So you may be asked USB type 3 apparatuses, which out of the following? USB type 5 apparatuses, which out of the following? So you can better learn these, memorize these. Based on IP as well, we have paddle and basket. Based on BP as well, we have basket, paddle, flow through, cell. So thank you so much. We will meet in the next session.